Hey everyone, how you doing? We just finished doing our play posit for Punnett Square, so you should have a little bit of an idea of what that is and how it's done. And this is just a prediction of what the outcome from two parents would be. So if we look here, we have Punnett squares are a way to predict the proportion. The proportion is also the amount of, or the percent of, possible genotypes in offspring. So offsprings means baby. So what we do is you make this square, right? So I'm going to put one parent's genotype on the top. So this is it. So this A and little a, big A, little a is heterozygous, right? And then I have this other parent, which is little a, and that is homozygous recessive. So how we're going to do this is we're going to bring down this big A here and then bring over this little a here, and that's going to give us big A, little a. So all we're doing is intersecting these. So the next one would be big A is going to come down to this bottom one. Little a is going to come over. And here I'm going to have, again, big A, little a, because I have a big A from here, little a here, we intersect here. Then over here I have my little a coming down, and I have this little a coming down. So this is going to give us little a, little a. All right, so now we have this last square, and we're going to intersect them, right? So we're bringing our A down here, we're bringing our little a over there, and we now have little a, little a. So you can see we're just doing these intersections to fill in the squares. And this one will code for just a single trait. There are ones that you do bigger that code for more than one trait, but we're going to learn this first. Alrighty. So next we have, and you should be filling in your notes for this. Move this up. So we're going to talk about crossing a heterozygous plant. Remember, heterozygous is two different traits. So A is dominant and little a is recessive. So when I have my parent pea plants, they're both going to be heterozygous, right? So they're going to be big A, little a. And that's going to give us yellow. So what that can kind of tell us is that if my phenotype is yellow, this capital A must be yellow, right? And then if I look down, my little a is going to be green. So crossing two pea plant, I can't talk today, pea plants that are heterozygous for seed color, big A, little a, like we see here, will produce offspring, and offsprings are babies, into this ratio. And we're going to look it down here, but we're going to first see how this works here. So I'm going to bring my big A down here. I'm going to bring my big A over here. So remember, I'm still doing that intersection of where they go. So I have big A, big A. And since I know the phenotype for the dominant trait is yellow, I have this yellow circle here. Okay, next I'm going to bring this big A down to the bottom here, and I'm going to bring this little a over here. So again, they're intersecting at this, and I have big A, little a. And my dominant trait is yellow for that big A, so that's why I have a yellow P here. And then we're going to go here. My little a comes down, and my big A is coming over. So this is giving me that heterozygous trait again, which since my capital A is dominant, it will be a yellow P. And then we have this one down here. And I bring my little a down here, and I'm going to intersect with this other little a here. And this is going to give me two little a's, right? So that's homozygous recessive. So only when it's homozygous recessive is that color going to show up. So the green, this would be green because it codes for, the hidden trait in here is green too. So if we look at each one of these, we have 1/4 is homozygous dominant, right? So that's this one here, homozygous dominant. It has a AA. Then we have 2 or 2/4, right? We have 2 that are heterozygous. So I have this one here and I have this one here. And then I have one here that is homozygous recessive, meaning I have 2 little a's. So I have 1 homozygous dominant, 2 heterozygous, and then 1 homozygous recessive. So you can think of this as each square is 25 cents. Like let's say this whole thing was $1, right? So when you're doing percents, this might help you make sense. So I have 1/4, right? Or 25% of big A, big A. Here, since I have 2 of these, I have 2/4, right? Which is 50 cents. So I have 50% of heterozygous. And then again here, since I have 2 little a's, I only have 1 of them. So that's 1/4, right? So 25 cents. So it'd be 25% are homozygous recessive. So if we look here, so I have 1/4 that's AA. I have 1/4 that's little a, and I have 2/4 that are big A, little a. And here's my little, wish that would move, my little key of what it is. So I have green plants are recessive and yellow plants, which are dominant. I'll give you a minute to catch up. So you're going to be doing a lot of things with the monohybrid cross. Okay, next we have what are called non-Mendelian genetics. So this is something that goes a little bit against the grain. So there a little differences. So we have some alleles that are neither dominant or recessive. And many traits are controlled by multiple alleles or multiple genes. So one of these is skin color, because you notice how people aren't, just black or white. We have varieties in between, right? So this is controlled by multiple alleles. So the first thing we're going to talk about is incomplete dominance. Incomplete. So this is where one allele is not completely dominant over the other allele. And what's going to happen is they blend together. So my dominant allele is like, hey, dude, I just don't have the strength to do this. So let's just blend together and we'll go there. So that's like I have this red flower here, which is a dominant color. And I have this white here and it's recessive. But when I cross them together, I'm going to get pink flowers. And this is called incomplete dominance. So you're only gonna, if you see this on the test, they have to tell you it's incomplete dominance, or otherwise you would do it the other way, where you have a dominant and a recessive trait. It's only if they say incomplete dominance and they give you a cross, and then it would be blended together. Okay, so here is another example of incomplete dominance. And we have here, we have here, this W goes, we're bringing them down, right? This R comes down, the W comes down, the R comes down, the W comes down. The R comes down, the W comes down, the R comes down, the W comes down. So these were all red flowers, completely red flowers, and these are all white flowers. But with incomplete dominance, our white flowers crossed with red flowers are going to equal pink flowers. So you can see here, and also with this little diagram here, this Punnett square. So this would be your genotype here, and this would be your phenotype here. Okay, and then we have one more, and let me move this. And this is called codominance. So this is where both alleles contribute to the phenotype. So, and phenotype, remember, is what that organism actually looks like. So both alleles here are expressed or shown. All right, so in incomplete dominance, we blended the colors together, and in codominance, they're both like, I'm in charge, no, me. So they're going to both be expressed. So if we look at this cow up here, we see it's a red cow, and I'm going to indicate that by capital R, little r, capital R, little r. And then over here, I have this cow, and he's a white cow, so he's going to be capital R, little w, and capital R, little w. So the small letters are telling us what the color is. So when we cross this, we're going to bring down this capital R, little r here, and then we're going to bring over this capital R, little w. So that's red and white. So if we see this cow, it has both red and white markings on it. So that is codominance. So you're going to have it, since these are both, if this is homozygous and this is homozygous, every one of these squares is going to be the same. So we're going to have 4 spotted red and white cows. Nice. And, oh yeah, here you go. So it would be capital R, capital R, W. The little letters are standing for the colors. So we would have red and, white spotted cows. And that's it. So we're now going to go on and do a worksheet. All right, bye.